by the Universitas Indonesia Visiting Professorship. So to short the time, I would like to invite Prof. Septilia Inawati as director or the chief of the biomedical science program in Universitas Indonesia to do the welcoming speech. Please, Prof. Ina.
Okay, thank you very much, Dr. Yordan. Assalamualaikum warahmatullahi wabarakatuh. Very good morning, ladies and gentlemen. I hope you all staying healthy and safe in this pandemic situation. First of all, I would like to welcome the honorable guest as our visiting professor, Professor Tomoyoshi Nozaki from the University of Tokyo, and also the honorable speaker, Professor Agnes Kurniawan, PhD, from the Department of Parasitology, Faculty of Medicine, Universitas Indonesia. On behalf of the Master Program in Biomedical Sciences, Faculty of Medicine, Universitas Indonesia, I would like to express our sincere gratitude to both honorable speaker for sharing us your expertise in parasitic disease that we will discuss in this forum today. E-Forum Biomedica is a regular scientific seminar held by the Master Program in Biomedical Sciences, Faculty of Medicine, Universitas Indonesia, for our students and also for all the biomedical scientists in Indonesia in general. Today we have about 293 registered participants. And we also open this forum electronically through our YouTube channel. I see that there are so many participants that are enthusiastic in learning this topic today. The MPBS FMUI, Master Program in Biomedical Sciences, was established in 1979. Our program is the first graduate program in Universitas Indonesia, and for the biomedical science program, also the first in Indonesia. Our vision is to be one of the leading biomedical education center in Southeast Asia, focusing on translational research. After assessed by the ASEAN University Network last year, this year we received a funding from Universitas Indonesia for the international academic reputation through several collaborative programs, such as the Visiting Professorship Program and also International Benchmarking Program. We are very grateful that Professor Nozaki has accepted our invitation to be our external reviewer for the International Benchmarking Program. We also plan to have a meeting with Professor Nozaki for this program. And also we can have another meeting for the collaboration between University of Tokyo and Universitas Indonesia in the next future. I hope that the involvement of Professor Nozaki through this program could enhance and broaden the collaboration between UI, Universitas Indonesia, and University of Tokyo to establish the standard, international standard quality of master graduate program in biomedical sciences at our faculty. To all audiences, thank you for your time and have an inspiring insight and fruitful discussion from both speakers. Enjoy this and fruitful discussion. Have a good day.
Thank you very much, Prof. Ina, for your invite for your introducing speech. So to make it shorter, so we would like to start this. So it will be moderated by the Dr. Wawaimuli, and also I would like to tell the to inform the for the audience to switch on turn on the your video, but please turn off your audio or speakers. So now we would like to start. Is there any something, Prof. Ina?
Picture.
Hello.
Can we have picture together with the participants?
Oh, okay. Okay, so before we start, we also would like to take a picture for the all of the audience. So please turn on your video right now, and please, Adin, could you help us to do the picture session?
Yes, I will take 3 pictures. So the first picture, ready, 1, 2, 3. And then the second picture, 1, 2, 3. And then the last picture, 1, 2, 3. Okay, thank you.
Okay, thank you very much, Miss Adin. So is there any something to inform again? Prof. Ina, is Ina?
It's okay.
Okay, okay, let's start. So we start the forum, and then please, Dr. Wawaimuli, to do the starting point. Okay.
Okay, thank you very much, Dr. Yoda, for nice introduction. So hello everyone. My name is Wawaimuli. I'm from Department of Pharmacology, Universitas Indonesia. So I will be the moderator for this session. So good morning, very good morning to everyone. Assalamualaikum warahmatullahi wabarakatuh. First of all, I would like to welcome you all to this presentation. In this special occasion, we would like to have a presentation about drug discovery in parasitic disease. So before the presentation begins, I would like to introduce our presenter. So there will be 2 speakers in this webinar session. The first speaker will be Professor Agnes Kurniawan from Department of Parasitology FMI, followed by Professor Tomoyoshi Nozaki from University of Tokyo. So after completing the 2 speakers, we will have a question and answer session about 20 minutes. So, all right, without any further ado, I would like to welcome the first speaker, Professor Agnes Kurniawan, to deliver her presentation. But before Prof. Agnes deliver the presentation, let me share her CV. So, Adin, may I ask you to share the CV of Prof. Agnes? Adin?
Ya, sebentar.
Okay, Prof. Agnes is a professor in medical parasitology. So he is graduate from University of Indonesia in 1987, and then also a PhD parasitology from in 1996. And then Agnes also have a related to the toxoplasma and the other parasites. So without further ado, I would like to invite Agnes to this.
Okay. Yeah. Thank you, Dr. Lee. Can you hear me clearly? Hello. Can everybody hear me clearly? My voice clearly?
Yes, Prof.
Okay.
Yes. Yes, Prof.
Yeah, the network is not so good. Yeah. Okay. Thank you, Dr. Muli, and good morning, everybody. Thank you to Prof. Ina, and to Dr. Dan, and to our honorary speaker, Prof. Nozaki. Morning, everybody, and so I will share my presentation. Okay, on intestinal infection in Indonesia. Yeah, there will be several studies I will present to intestinal infection. About what is how is the profile of intestinal the children in Jakarta, and about the profile of adolescent and in South Sumatra, and then the glimpse of the profile of intestinal parasitic infection in particular among the affiliates study from itu hospital Jakarta and lastly I will talk about the issue that's protocol with okay so the protocol among primary school children this southern part of Jakarta. So this the 56 was we found 1.9 harbor see yeah last time there's also mixed infection small under information and So there is good so first highly preferred or the and we didn't find a single it's very this patient.
Check your microphone, Prof. Agnes. Can you check your microphone? We cannot clearly listen to your presentation.
Yeah.
So maybe you should more close to your microphone.
Okay.
Thank
Hello.
you. Now is it more better? Okay, this good. Yes, it is better.
Okay.
It is better now. Okay, thank you, Prof. Agnes. Sorry.
It is better. Okay, thank you. So we can, I continue, yeah. Okay, then we continue with the study with typing of the blastocystis, because blastocystis is still was still regarded as either, so there is a confusion, yeah, whether this is pathogenic or non-pathogenic. And then at that time, we try to associate whether this the presence of blastocystis is associated with the symptoms. So we asked the history of the diarrhea and other intestinal, gastrointestinal symptoms, yeah. And then this is the result we found that we identified 4 subtypes of blastocystis. Actually, there are so many subtypes of blastocystis, and then this parasite presence not only in human but also in various animals. Yeah, start from the small one like the rats, and then up to the pig and cat and dog and so on. Yeah, so among the sorry, I'm sorry. So among the study population, we found 4 subtypes of blastocystis. That is subtype 1, subtype 2, subtype 3, and 4. We know that there are 9 subtypes commonly found in human, but in our population, we found only the subtype 1 up to 4. And then the dominant one is the subtype 3. And then we did the analysis, yeah, and then it was found that the subtype 3 was the dominant one, and it was significantly associated with the symptoms, yeah. And then this is genetic analysis, yeah, where the subtype 4, which was reported, yeah, prevalent among the rats in Sumba, yeah, that is in East Nusa Tenggara, only found in 1 student, yeah. And then another study about blastocystis among this among the patients, yeah, with HIV, yeah, the intestinal protozoa among the HIV, we observed that there is 70.2% of the patients Yeah, the number of patients at that time was 318, yeah, with chronic diarrhea, and 72.4% harbor intestinal parasites. And then we saw here that there are various parasites, yeah, from blastocystis, which was again the dominant one, yeah, and then cyclospora, cryptosporidium, giardia, and then an amoeba histolytica or dispar, and also a small number of worms that was Ascaris lumbricoides we found. And then this is the prevalence, yeah, of the parasites among the HIV/AIDS, yeah. And then again, we found that the blastocystis is the dominant one, and then it present with either a single infection or mixed with either cryptosporidium, giardia, and other intestinal protozoa up to Isospora belli, yeah, or Isospora hominis, yeah. And also there is an opportunistic parasite like Cryptosporidium, Isospora as well, yeah. So this blastocystis pathogenic parasite are not pathogenic because we found in the community there is high prevalence, but some with symptoms, some without symptoms. So we continue study with this one, yeah. So we look at the population, yeah. This is among the adolescents in Palembang, South Sumatra, where we do on IBS, yeah, the adolescents, that is the high school students in Palembang, and those with irritable bowel syndrome, yeah. We look at whether there is blastocystis infection or not, and then the number of students we selected was 454 students, and from there we put into the groups between IBS and non-IBS groups, and then we check for the blastocystis, and we do also the typing and try to associate it with the type of IBS, because IBS can be mixed type or unclassified or constipation type, yeah. And then, so again, yeah, the presence of blastocystis is quite high, up to 30%, yeah, among the adolescents. And then, so among IBS subjects reach up to 36.5%, while among the non-IBS, that is 28.6%. And then we identified 3 subtypes in this population. They are subtype 1, subtype 2, and subtype. And then the association with the IBS type, like this, yeah. So among those population, yeah, we see there is IBS with diarrhea symptoms, and then IBS with constipation, IBS with mixed symptoms, and IBS unclassified. And then we see here that most individual, yeah, with subtype 1 presented with IBS diarrhea, while the rest they are distributed, yeah, like this. And then the association, when we check by statistical analysis, it was significant, yeah. And then from here, we conclude that blastocystis subtype 1 is a pathogenic one and more likely yeah, to develop the IBS type of diarrhea, yeah. So those with diarrhea symptoms, yeah, they are profound, yeah, favor 2.9 times, yeah, to develop IBS diarrhea. So those with subtype 1 is associated with IBS diarrhea type. And then a little bit on amoebiasis, yeah. So I would like to report about the problem of amoebiasis in parasitology laboratory at Department of Parasitology, yeah, because Department of Parasitology, yeah, receive the specimens, yeah, from the hospitals and also from the not only from the Cipto Mangunkusumo Hospital, but also from other hospitals in surrounding in Jakarta, yeah. So there is examination about the serology amoeba, yeah, and then it was 62 samples examined, and then from the 62 samples, we can track, yeah, 33 samples could be traced from the medical records in Cipto Mangunkusumo Hospital, while the rest from other hospitals outside Cipto Mangunkusumo. And then from this 62, we found 27.4% positive for the serology. Yeah, so that means they are positive for the amoebiasis. And the clinical manifestation and symptoms mostly associated with 25% with liver abscess, and then some with 25% of the liver abscess, they are with manifested with pleural effusion as well. And then the seropositive result, yeah, mostly from cases with liver abscess, yeah. And then most of the patients, yeah, examined, they have got metronidazole treatment before the serology examination, and also the clinician did aspiration for their abscess. And then metronidazole was the drug of choice, which is commonly used for the treatment of liver abscess. And then recently, yeah, about 3 years ago, there is an issue about resistance to metronidazole in amoebiasis. So there was a report from the pediatricians that they have treated the patients diagnosed as amoebiasis, and then those patients, the children with chronic bloody diarrhea, and the children have given the metronidazole, but on the evaluation of the feces, they still found ada kasus yang Halo.
Sorry for the interrupt. Please continue, Prof.
Okay. Okay, yeah. I will continue. Okay. Yeah. And so on evaluation, on the fecal examination, yeah, the laboratory reported that there's still an amoeba histolytica cyst found. So the pediatrician was wondering, yeah, whether they are confused whether this is a type of resistant an amoeba histolytica due to in metronidazole, yeah. And then so they sent us the fecal specimens, and then we reported here that there are 10 cases, yeah, from children, and sent to our laboratory and diagnosed as colitis or suspected for amoebic dysentery. And then we did this examination, yeah, with the direct and the concentration, and then also staining with the trichrome staining. And then we found that those diagnosed as amoebiasis resistance to metronidazole, actually it was the amoeba fragilis, yeah. And then another 5 cases, it was the amoeba fragilis mixed with blastocystis or an amoeba histolytica dispar infection. But also there are some which was not found, yeah. And then in these cases, yeah, we asked them to send the hot stool because the Amoeba fragilis is very fragile and easily died. And then it needs a very fresh stool. So that's why it's not easy to find the parasite, yeah. And then this is the profile of the patients, yeah. The patients from their babies, yeah, less than 2 years. And then also there's one female patient, 65 years, have been treated several times with metronidazole, but failed. And then the symptoms again and again, yeah. And then we found that she suffered from blastocystis and also the Amoeba fragilis. And then this another adult patient with diarrhea, also with blastocystis and the Amoeba fragilis. So, so this is the parasite, stained with trichrome, yeah. And then, so the treatment, yeah, the treatment for the pediatric patients less than 8 years old, they were treated with either paromomycin, while the older and adult patients, they receive doxycycline or paromomycin or the macrolide. And the pediatric patients, apart from paromomycin, they also get the macrolide, that is either erythromycin or other macrolide, yeah. And then the therapy resulted in improvement of the patient's condition. However, the issue of metronidazole-resistant amoebiasis needs further investigation. Yeah, so in conclusion, that from these several reports, yeah, we observed that intestinal protozoa infection is still high prevalent among the Indonesian population, even though many studies reported about the intestinal worm infection, the STH, but the intestinal protozoa infection still lack of reports and lack of studies, yeah. And what we reported here, that is in Jakarta, and I'm sure that the prevalence outside Jakarta in rural area may be even higher, yeah. And also if the amoebiasis in the community mostly stay as cryptic infection, and then which manifested later as liver abscess with or without pleural effusion. So if in our examination from our laboratory, reported 25 up to 25% of patients with the on the serology, yeah, they suffer from liver abscess with due to amoeba. So I'm sure that the prevalence in the community should be higher. However, it was stay as a cryptic infection, asymptomatic or with symptoms, but then the symptoms then diminish, yeah, quickly. And then, or maybe disregarded or by the patient, and then the patient either got treatment by themselves or they don't check for the fecal examination, and then the doctor gave the treatment as usual. Yeah, and then blastocystis is still the predominant and also commonly found intestinal protozoa on fecal examination. However, due to many types of blastocystis, of course, we should be it should be borne in our mind that several types like subtype 1, yeah, or subtype 2, which is dominant, can be associated with the symptoms, yeah, and this, of course, may need treatment. And then the Amoeba fragilis is an emerging protozoa infections, which is difficult to diagnose, and it's not susceptible to metronidazole treatment. And then this parasite may misdiagnose as an Amoeba histolytica because because of the morphology and also which we rely on the morphology examination. Okay, and these are all the contributors for the studies reported, yeah, from our lab, and also all the biomedical students, several biomedical students, and also some clinicians from the pediatrics, from the HIV clinic at Cipto Mangunkusumo Hospital. So that's all, and thank you for your attention.
Okay, thank you very much, Professor Agnes, for the nice presentation. So Prof. Agnes already deliver related to the current situation of intestinal protozoa in Indonesia. So she already talked about the epidemiology and then frequency, and then the most important is the issue related to the drug resistance in some protozoa. So now we will moving to the next presenter, our honorable guest speaker, Professor Tomoyoshi Nozaki from Department of Biomedical Chemistry, University of Tokyo. So before he start his presentation, may I share his CV? Adin, could you please share his CV? Okay, Professor Tomoyoshi Nozaki, the current affiliation is Department of Biomedical Chemistry, University of Tokyo. So he was graduate from School of Medicine, Keio, University Tokyo in 1987, and then graduate from School of Medicine, Keio University to get a PhD degree in 1997. So actually, I have already searched his main major is related to the parasitology. So he published a lot of publication, more than 200 publication, and each index is 41. So it is very amazing. So now times is yours, Professor Tomoyoshi Nozaki.
Okay, thank you for introductions, and it's really honor to be a guest speaker at your prestigious faculty of Universitas Indonesia. Can you hear me all right? Because I can also hear my voice echoing.
Yes, we can hear you clearly.
I think something wrong because I can hear the echo.
It's clear for us, Sensei.
Can I continue? It's clear.
Yes. Yeah. Continue.
Okay, so I continue. All right, so I will share my screen first. Can you see? Hi, can you hear me? Can you see my screen all right?
Yes, Sensei, it's clear.
Okay, all right. So I'll start. Okay, so since the time is limited, I will jump onto the topic of the presentation I have today. The topic is drug discoveries against infectious diseases, mainly on parasitic infections, by utilizing bioresources from this country, from your country, Indonesia. And Indonesia is famous for its mega biodiversity. You have a huge land with lots of different climates, different geographic characters. So I have been collaborating with Indonesian researchers such as BPPT, LIPI, Universitas Airlangga for the last 7 years in this project. So I'm going to summarize what we have been doing and what perspective we have in the future. And I really hope that the Universitas Indonesia can also join us, and we can create a new liaison and maybe research network on this topic in coming years. And so this is the content of my talk. I just to give you a brief background of malaria and amoebiasis, the two major diseases we are dealing with at this moment. But currently we have also started working on drug developments against tuberculosis, as well as dengue fever. I know that both of the diseases are also a major concern of Indonesian people. And we're gonna give you the example of drug developments using Indonesian microbial resources. And finally, I will touch upon another branch and leg of anti-malaria drug developments using a chemically defined compound library from Drug Discovery Initiative, University of Tokyo. Maybe I don't need to rephrase the importance of malaria. I think all the medical students, as well as the students in your faculty at the Universitas Indonesia, knows about the significance of the disease. Malaria is endemic in close to 100 countries worldwide. Mainly, major infections occurred in Sub-Saharan Africa, Saharan Africa. And the more than 200 million people infected annually, causing over 400,000 deaths annually. And of course, it is the most important protozoan cause of mortality. And at this moment, there is no established available vaccine available, although RTS-SA01 from GSK, GlaxoSmithKline, finished the phase 3 trials, and the result was somehow good partial protections. But to be honest, it's quite disappointing because it has only a partial protection effect, and it's not really at all to our satisfactory levels, in, giving enough protections against new malaria infections. And artemisinin combined therapy, ACT, is commonly used for Plasmodium falciparum infections. But again, the drug resistance, not only to the pre-existing drugs such as SP, cefodoxime, pyrimethamines, as well as mefloquines, chloroquines, as well as new lines of artemisinin derivatives are emerging. And in 2010, 10 years ago, close to 30% of cases, malaria cases, are resistant to dihydroartemisinin, piperakines, and artemisinin combined chemotherapies. And even for less malignant malaria, P-vivax infections, more than 20 countries chloroquine resistant has been developed and reported. So both vaccine development as well as drug developments with a new mode of actions and new chemical entities are urgently needed against malaria. And I learned today from the previous presentation by Professor Agnes Kurniawan that amoebiasis also poses a serious health threat and social problems in Indonesia, although the current status of amoebiasis seems to be quite cryptic and understood. But I can see the impacts of amoebiasis in Indonesia as well. And amoebiasis roughly estimated to affect approximately 1% of the whole population on the planet. And this is a deadly protozoa infections and only after malaria. No vaccine available. And as explained by Professor Agnes, metronidazole is the drug of choice. and virtually only drug available against metronidazole. The paromomycin has been used against cyst carriers, asymptomatic patients, but it doesn't have trophozoid killing activity. And metronidazole has a lot of problem because it's a calcinogenic and tetragenic, and it cannot be applied to either pregnant women or children. And resistance reported in giardiosis, trichomoniasis, and also Helicobacter pylori infections have proven already a drug resistance against metronidazole can be easily acquired by bacteria as well as protozoan parasites. So again, we need a new drug against amoebiasis. So I want to start my presentation with this slide. A key of success of drug development is a partnership between countries and between academia, public sectors, as well as private funders. Money is really required, and literally huge money is really needed for drug development, drug and vaccine development. We are fortunate that we have been carrying out the two major projects on the drug developments against malaria and amoebiasis through two major partnerships. The first one here shown bi or trilateral international collaborations between academia, university, research institutes, as well as private companies. And we found funders from Japanese governments, such as Japan International Cooperation Agency, JICA, as well as Japan Agency for Medical Research and Development, abbreviated as AMED. And this program was started like 7 years ago and was completed March last year. And I'm happy to announce that this 5-year project was terminated, but renewed for additional 6 more years, which started last April 2020. So the second phase of this bi or trilateral international collaborations between supported by JICA and AMED, and also Indonesian governments like a BPPT, LIPI, as well as RISTEC, will be continuing for next 6 years. And second project I want to touch upon is a private-public partnership. And big funder in this case is the GHIT Fund. It's a Japan-owned private funder. Its abbreviation of GHIT is written here. It's called Global Health Innovative Technology Fund. Half of the fund for this NGO, non-governmental organization, is from Gates Foundations, and 1/4 is from Japanese ministries such as AMED, Ministry of Health, Ministry of Foreign Affairs, and 1/4 come from Japanese pharmaceutical industries. So we could take advantage of this funder, GHIT Fund, to accelerate drug development through a public-private partnership, PPP. So I'm going to start a presentation on the first topic. It's the AMED and JICA supported bilateral international collaborations. So the justification we started project with Indonesia is multifold. One, I had learned from my Indonesian colleagues when we started the project 6-7 years ago that Indonesia, the materials needed to produce drugs which is consumed in the countries, like 90% or more than 90% of the drugs are mostly imported. And there is no sufficient infrastructure to produce domestically the new drugs, for instance, for communicable and non-communicable diseases. And even for the antimalarial chemotherapies, I heard, I don't know how true that is at this moment, but the very low availability of standard ACT chemotherapy for malarial patients. Only 30% of cases are treated appropriately with the ACT. So we proposed 7 years ago that we're going to propose the new project by combining Indonesian bioresources and Japanese technologies and expertise on the drug developments from microbial resources. As you might know, that the Professor Satoshi Omura from Kitasato University, who is a Nobel laureate 2015 for physiology and medicine. And as represented by this example, Japanese university and industries in general have a lot of advantages on utilizations of microbial resources as a source of drugs. So combining this expertise with Indonesian rich microbial resources, we thought we can create a new platform for drug developments. And once we make it, then we can utilize a platform for not only parasitic diseases, but also for a panel of infectious diseases, as well as like a cancer research, maybe Alzheimer's diseases, diabetes, and other important diseases in the country. So, so this is a banner for the projects when we started it. Our major counterpart was the Biotech Center, currently called Laboratory for Biotechnology at BPPT, which is located in SpParK area. And we also have a counterparts and partnership with LIPI, INDACC, Indonesian Culture Collections, located in Cibinong, as well as the Airlangga University Institute of Tropical Disease at Airlangga University, Surabaya. So for this project, we started from scratch, basically. So we set up the missions to collect microorganisms such as fungi, actinomycetes, from remote islands in Indonesia. So we did it for 5 years and collected up to 3,000 new microbial organisms and made libraries and store them at BPPT. And we also utilize plants, but the very small in number. Mainly we concentrated on the microbes, microbial organisms, and we made extracts, and we made approximately 20,000 extracts from fungi and actinomycetes, and we use them for the drug screening. So this is a homepage for the SATREPS. It's abbreviation for Science and Technology Research Partnership for Sustainable Developments. If you are interested, in what we did, what we accomplished in the past, please search by Google by typing SATREPS and my name Nozaki. I think you'll find the necessary information. The project is terminated last March, but it's gonna be renewed. It has been renewed already for the next 5 years. So aim of the project was to identify microbes that produces metabolites and secondary metabolites mainly that inhibits enzymes from Plasmodium falciparum and Entamoeba histolytica, or identify the metabolites which inhibits the growth of the parasites by screening Indonesian microbial extracts, and purify the active compounds and determine their structures. That was a goal of our project. So again, we set up the missions to harvest microbial resources. We made culture, and we had established a screening system in Indonesia, both for enzyme-based assay as well as cell-based assay. And once we identify hits, we scale up the culture from like a 4 ml culture to 1 liter, 500 ml, 1 liter culture using a big Erlenmeyer flask. And we purify the compounds using TLC, a thin layer chromatography, or HPLC, high performance liquid chromatography, as well as open column chromatography. And finally, we tested those candidate compounds using small rodents such as hamsters and mice. And we identified them as a potential leads for fight. And we are going to modify the chemical structures and try to improve efficacy and decrease the toxicity to human cells, and double check and validated the safety for the animal use. So chemical major outcome, right? I skipped this slide. So major expected outcome of such activity is multi-layered again. So for Japan side, I think our government emphasized importance of medicine derived development of Japanese economy. So only help us to sort of propose a new platforms via bilateral collaborations, and this perfectly suits the basic plans of science and technology of Japanese government. And this also, we believed that our project also suits Indonesian size governments. This help Indonesian researchers and industries to establish knowledge and infrastructure needed for lead discovery against infectious diseases, which can be used for non-infectious diseases in coming years. And utilization of its your own bioresources in these mega biodiversity countries. And this nicely fits your master plans for accelerations and acceleration of Indonesian economic developments 2011 and 2025. It's called MP3EI. I think this is giving information from my colleagues from Indonesia. I hope it's right. So as I said, so we had 4 partners in Japan and 3 partners in Indonesia. And I just very briefly touch upon the outcome of our major achievements. For anti-amoebic lead compound discoveries, we started with 7,000 microbial extracts and identified 30 hits, very potent hits. And out of and those 31 is like a 22 fungal extracts as well as 9 actinomycetes culture extracts. From this 22 fungal extracts, we purified and identified 1 compound called fumagillin and the other compound called ovalicin. And these 2 compounds are very similar in structures. If you just look at this structure, this is a cyclohexane ring with 1 epoxy side attached to the ring. And also on the side chain, there is another, the other epoxy side. So this is called a spiral epoxide cyclohexane basic structure. And this structure is very well conserved between ovallicins and the other compound fumagillin. And fumagillin differs from ovallicin with this extension. Okay, and they are identified and purified from different fungal species. But we learned later that the fumagillin and ovallicins are quite common secondary metabolites from fungi. So it's not us who first identified this activity against anti-amoebic, but it was discovered 70 years ago. It's a shame to say it, but this is a reality. It was identified, fumagillin was reported 70 years ago, even tested in human, and to be proven to be partially effective against amoebiasis patient in Egypt. Okay, since then, for some reasons for poor efficacy, as well as some adverse effects, drug developments starting from fumagillin and ovallicin seems to be ceased. But last 20 years, fumagillin and ovallicin are being started, developed again as the angiogenesis inhibitor, anti-cancer drug, as well as obesity drugs. And in France, fumagillin is used as anti-microsporidia drug in HIV patients. It was registered already. And in the US, now the fumagillin derivatives is developed as anti-obese drugs in the patients who has a genetic disorder, neurological disorders, who tend to gain weight. So those facts actually endorse the safety of fumagillin-derived compounds. So we are going to start further chemical modification of fumagillin and ovallicin in coming years. So ovallicin and fumagillin is a really amazing drug. This is a metronidazole. This is IC50. This effective concentrations, effective dose, which kills entamoeba histolytica cells in vitro. And IC50 value is 2.5 micromolar. Okay, 2.5 micromolar. And the ovallicin and fumagillin show the IC50 of 20 or 70 nanomolar. So this is 100-fold smaller than IC50 value of metronidazole. So in other words, ovallicin fumagillins are 100-fold more potent against entamoeba, at least in vitro. And safety was also verified using human non-cancerous cells, such as the fibroblast MRC5. Again, this is a pretty safe drugs, and selectivity index is over 4,000. So ovallicin seems to be very safe and potent compound. And this was also verified by animal model. We use hamster liver abscess model, so we directly inject amoebic trophozoites into a left lobe of the hamster and sacrifice the animal 6 days after inoculations and weigh the volumes, measure the weight of the abscesses in the whole liver. Okay, and we prove that the like a 1 milligram per kilo subcutaneous inoculation of ovallicins completely cure amoebic liver abscess. And target of fumagillin and ovallicin has been demonstrated already. And this is methionine aminopeptidase 2, MET-AP2. And MET-AP2 is involved in the removal of methionine, amino acid methionine from newly synthesized nascent polypeptide chains during a post-translational modifications such as lipid modifications at glycine at the carboxy terminus, like lipidations on the nascent newly synthesized the peptide. And the MetAP2 is well conserved from bacteria to human, but the human has two isotypes such as MetAP1 and MetAP2, whereas entamoeba and Plasmodia has only MetAP2. This can validate why MetAP2 inhibitors can be validated drugs. Okay, I forgot to mention that antisphagilin and ovalicins are not only effective against amoebiasis, but this also effective against malaria. This is also effective against Trypanosoma brucei, and also it's also effective against Giardia intestinalis, which Professor Agnes presented prior to my presentation. So fumagillin and ovalicin seems to be very attractive lead compounds for the further drug developments. So this is just, again, a summarize what sort of compounds we identified other than fumagillin and ovalicin. We also identify citrinin, and the carboxylated citrinins are from different islands of Indonesia. And we also targeted enzyme, amoebic enzyme, and we identified a few compounds, but I skipped those slides for the sake of time. And for the discovery of the inhibitors against malaria parasite, Plasmodium falciparum, cell-based screening yielded this compound called borelidin. This is, as shown here, this is a macrolide antibiotic. It was isolated from streptomyces. And originally this was named, borelidin was named after the activity against Borrelia. And it was shown 10 years ago that borelidin inhibits the growth of erythrocytic stage parasites of Plasmodium falciparum. And the target is also known, and borelidin inhibits, again, translations. It's called threonine tRNA synthetase, again, involved in nascent polypeptide change. We also targeted few enzymes essential for mitochondria respirations in Plasmodium, such as DHODH, dehydroorotate dehydrogenase, as well as the related respiratory enzymes such as NADH, oxidoreductase, NDH2, and also MQO, malate quinone oxidoreductase. But for the sake of time, I skip those details. And we identified using DHODH as the target enzyme. We identified more than 10 active compounds and inhibitors of the enzyme, which partially show the cytotoxicity against malaria parasite. The compounds are shown here, such as gentesyl alcohol and mitomycin C, altemucin, mitorubrinol, and mitorubrinolic acid. And unfortunately, all of those compounds are not new in structure. And some are known to be antimalarial, and some are known to be anti-cancer compounds. But we believe that our approach using Indonesian microbial extracts as a source for the identification of new compounds with the pharmaceutical values, I think we could provide the proof of concept of our approach. Okay, so again, this is a summary of the antimalarials. And so we identified DHODH, MQO inhibitors, as well as anti-Plasmodium falciparum proliferative compounds from 4 major Indonesian islands. Right. So in summary, we identified several major lead compounds that have a potential for further developments as anti-amoebic and anti-malarial drugs through this bilateral collaborations called the SATREPS program, which combined advantage of our two countries. And we produced and created a microbe library of the 20,000 from Indonesian bioresources and utilize the library for screening. And technology and knowledge are needed for microbial collections, identifications, archiving, as well as cell or enzyme-based screening methods, and also a structured elucidation such as NMR and mass spectrometries has been transferred successfully to Indonesian researchers. And at this stage, we proposed that two lead compounds, humagidine or valicin, as well as borelidine, as very promising lead compounds against amoebiasis and malaria for the next stage of drug development. And this is the second stage of our drug developments with Indonesian. And this time, we try to create a network a little bit larger. So we also invited Malaysian researchers. I know that Indonesian and Malaysia has a close liaison with the similar cultural backgrounds and educational systems. So, and they have expertise for the chemical synthesis. So we thought it's advantageous combining Indonesian bioresources and our microbial resources, as well as the Malaysians' chemical synthesis, chemical developments activities. So this time we proposed a tri-rational collaborations, and the major aims and objective of the study is chemical modification, structural modifications for optimization, as well as safety testing using both in vitro and in vivo models. And our partner this time we propose is the again, Laboratory for Biotechnology, BPPT, and Indonesian Institute of Science, LIPI, and the IPB University Primate Research Center, because we must use monkeys for the final validation of safety. And IPB University Primate Research Center in Bogor is the only institutions in Indonesia, as far as I know, in academia, who have equipment to do the research. But again, I really want to create the network and the consortium which we have been developing in the past, involving all the researchers interested in the drug and vaccine developments in Indonesia. And we are gonna, I want to invite Universitas Indonesia to join us for a part of the activity in the future. Okay, then very briefly, I will touch upon the second type of collaborations we have been doing, which is a private partnership. The major partner is the MMV, Medicine for Malaria Venture, which is the dry institution, which doesn't have any wet laboratory. So activity of MMV is to coordinate multiple centers for the drug development, including us, okay? And the major funder of the project is the GHIT Fund, as I said. So this is a private NGO, non-governmental funding agency. And instead of using microbial resources, we rely on chemically defined structure elucidated compound libraries owned by University of Tokyo. So this is the institutions called the Drug Discovery Initiative at the University of Tokyo, who has the over 200,000 structurally defined compound library with robotic collections and management systems. And they have, this is a pie graph of the over 200 compounds. Sorry. I think something wrong. Well, it's okay. It's okay. So the point I want to make is we are going to start. No, we started high throughput screening not on the 96 weight plates, but we use 384 weight plates using robotic system. So the throughput is quite high, and we can finish the screening of 200,000 compounds within maybe 3 months. So we
Sorry, Professor, your microphone is off. Your microphone. Okay, thank you.
Sorry. Can you hear me now?
Yes, yes, Professor.
Okay, so I have to repeat it. So we screened over 200,000 compounds from DDI, a drug discovery initiative, using 384 weight microtiter plates, which has a quite robust and high throughput, and we can finish 200,000 compounds, the screening of 200,000 compounds.
Sorry, Professor. Your microphone, excuse me. Your microphone, okay.
Why it's keep meeting? Because I'm not doing anything. Can you hear me now?
Okay, Prof. Yes.
Okay.
Sorry for technical problem.
That's fine. So we used phenotypic screening using erythrocytic stage parasites by a lactate dehydrogenase assay with a fixed concentration of 2 micromolar. And we selected only the compound which killed more than 90% of the parasites using the 90% cutoff point. And we identified close to 1,000 hits. And we further confirmed positiveness with independent growth assay called CyberGreen assay. With CyberGreen assay, this basically stained DNA in the plasmodium in the erythrocyte, again with a fixed concentration 2 micromolar. And this way we can sort of narrow down the hits. And this YDH-ODH, this is a bit complicated. Now pipeline of drug developments as for the antimalarials, quite many groups has been working on. And there are quite candidate compounds which inhibits DHODH, which is a mitochondrial enzyme of the plasmodium, is online in the pipeline. So we don't want to get another DHODH inhibitors, okay? So in this process, basically we, to make a long story short, we try to exclude any potential DHODH inhibitors. So which narrow down to 579 hits. And then we also tested against DD2, which is chloroquine and SP resistant cell line to make sure our new candidate compounds is also effective against drug sensitive as well as drug resistant cell lines. And after narrowing down from 200,000 to 500 or so, we determined inhibitory concentration of 50% IC50 for all the compounds. At the same time, we got the help from MMB, Medicines for Malaria Venture in Geneva, Switzerland, because they have a lot of experience. So basically, we try to remove all the overlapping fragments which are known to be antimalarials using MPO, multi-parameter optimizations. We also look for structural novelty using MMB owned database and also like a chemical database available. And finally, we also try to see whether or not newly identified compounds from DDI libraries are chemically attractive. In other words, no strange structures which likely causes side effects and difficult to modify chemically. So, and then we profile the actives. And we prioritize those candidates by life stage activity. In other words, we have to make sure the compounds are effective against erythrocytic parasites, as well as like a liver stage parasites, as well as insect stage parasites, like a gametocytes, oocysts, sporozoites. Okay, and we also want the fast killing drugs. So rate of kill is important. We want to have a fast acting drug. MOA is important, mechanism of action study, because we want to have the compounds which has a novel actions, novel mechanism of actions. Basically, we don't want to have any anti-forates. We don't want to have anti-ATP4, ATPase inhibitors, and we don't want to have another DHODH inhibitors for the reason I said. So we want to have a new compound with a new mechanism of action. ADME, absorptions, and the and metabolisms, and whole bunch of properties, pharmacological properties are also important. So after prioritizations from such parameters, we try to narrow down. And finally, we narrow it down to like 12 compounds at the bottom. And this is through whole the process I explained in 2 minutes, as well as finally, chemist eye. So we asked chemical directors, MMB, who has a lot of experience on drug developments in the pharmaceutical industries, to ask them, directors, do you like which compounds to be further developed in a hit to lead projects? So we finally selected 4 compounds and proposed hit to lead optimizations to GHIT fund. So finally, our proposal has been funded. The new it's called hit to lead optimizations of compounds, as well as the safety testing will be initiated this April, in 3 months. And in this new proposal, we highlighted 2 to 3 new series, apparently new scaffolds, new structures as the new antimalarials. And we are going to optimize physical chemical properties, and also try to improve efficacies and decrease the toxicity, and try to choose better pharmacokinetic and pharmacodynamic properties. Of course, we have to ensure the safety. Okay, so with this, I think, I hope that I can summarize what we have been doing in terms of drug developments against parasitic disease. But as I said, in this second phase of our SATREPS project, I talked 10 minutes ago that we also included tuberculosis, as well as dengue fever as a target disease. So if you have a particular interest in the drug developments against tuberculosis, for instance, at the University of Indonesia, I think I'm happy to discuss the further collaborations on the topic. And I didn't have a time to describe any of our research on amoebiasis, like epidemiology as well as molecular epidemiology. But before coming to University of Tokyo, I was at National Institute of Infectious Diseases, which is a referral centers for all the parasitic diseases. And I have also a very good liaison at the National Institute of Infectious Diseases, Department of Parasitology. So if you have a particular interest on like molecular epidemiologies of any parasitic infections, including toxoplasmosis, amoebiasis, giardiosis, cryptosporidiosis, etc., so I'm happy to do whatever I can do for the university and community. With that, this is my summary slide of all the participants at this stage and also in the past. As you might see, that I have quite many Indonesian students as well as a postdoc as my previous and current collaborators. And I don't know how many Indonesian students I have right now. I probably have 5 Indonesian 6 Indonesian students, 2 master students, 3 in PhD, and another one is coming from Brawijaya University from April. So I really hope that I can have a student from Universitas Indonesia, which is also very prestigious in your countries and who has a very good backgrounds of life sciences. With that, I would like to finish my talk by giving you this message, leave no one behind, which is a very important propaganda for SDGs. And if you are interested in the PhD program or even master program provided by MEXT, Ministry of Education, from Japanese government, as well as ADB, Asian Development Bank, please contact me. You can directly write me a letter, or you can ask coordinators of the university, Indonesian university, to contact me. But I'm happy to have you in my laboratory. I think with this, I'm going to finalize my talk. Thank you for your attention. I'm happy to take questions if you have any.
Okay, thank you very much, Professor Tomoyoshi Nozaki. So very nice presentation and so very nice invitation also, because of we have a lot of master student, maybe they will be interested to study in your lab. Okay, so now we are coming to the question and answer session. There are some questions from the chat box. So, okay. So may I read one question from Rivo Yudinata, Universitas Brawijaya. So the question for Professor Agnes as well as also for Professor Nozaki. So the question is related to the resistance of the Entamuba histolytica against metronidazole, even though it is a lack of clinical evidence. According to your opinion, what kind of possible mechanism that Entamuba histolytica can generate metronidazole resistance? So Prof Agnes, can you explain the possible mechanism of the resistance of metronidazole?
Prof, you're on mute.
Okay, yeah, thank you to Dr. Rivo. Yeah, regarding the pathway or the mechanism of metronidazole drug resistance, we do not study it, but that is from the literature said that is the pyruvate pyridoxine oxidoreductase pathway that is the role of this one. Yeah, That is the pathway that if we there is a synthesis, yeah, from the ferredoxin to ferredoxin oxidative to ferredoxin reduced, yeah. So if this was not disinhibited, then that will cause the resistance. But of course, I think Professor Tomoyoshi can explain better than me because he's working on the drug development. Thank you.
Sure. Yeah, a few comments to add. Although clinical evidence for drug resistance is not very heavy and very well documented, according to the clinicians who have experience on dealing with the patients, it's not many, but they sporadically experience the cases very hard to treat despite repeated treatment. So treatment failure is only one clinical evidence suggesting the presence of the drug resistance. But unless we isolate the strain from that patient, we cannot really formally prove the drug resistance and its mechanisms. On the other side of it, in the laboratory, it's quite easy to raise resistance. So you can cultivate amoebic trophozoites HM1 strain, for instance, in the presence of a small amounts of metronidazole. And as time goes, you can increase the drug concentrations. Okay? And you can easily make a resistance cell line. And by like a transcriptome analysis, such as RNA-seq or DNA microarray, it's been shown that some enzymes are overexpressed, such as PF4R that the Professor Agnes mentions, and also like flavin oxidoreductase, peroxyredoxin, and ferredoxins, and those enzymes and proteins involved in redox and anti-nitrosol defense seems to be somehow up or down regulated. But the overall picture of drug-resistant mechanism is not very well understood. Or other point is MDR, multi-drug resistant genes are somehow upregulated. But the causal connection is not very clear. So to answer the question that there are some evidence, there are many papers actually, but I think it's not really unequivocally demonstrated the main pathways of drug resistance. So this is an important point to address in the future.
Yeah. Okay. Yeah. Thank you, Professor Tomoyoshi. Because in our cases, we haven't proved it as metronidazole drug resistance because the patients that sent to us, it's not amoebiasis infection, but that is we found the Amoeba fragilis and also blastocystis. In which case, the Amoeba fragilis is not very susceptible to metronidazole treatment. And also like blastocystis, some may susceptible to metronidazole, but some may not. And there is also alternative drugs apart from metronidazole for treatment of blastocystis. Okay, that's all from me.
Okay, thank you, Prof. Agnes and Prof. Nozaki. So the next question also related to amoebiasis. So is there any sensitivity screening test for amoebiasis? Maybe before we start the treatment to the patient, is it necessary to do some kind of screening test to get a sensitivity test for each treatment, Prof. Agnes?
Okay, until now we haven't, we never do a sensitivity screening test for any parasites, except the malaria. We have the sensitivity screening test, but it's not regularly done, for malaria. But however, normally for the sensitivity screening test, we do it in vivo. That is the response to the clinical, to the drug treatment. That is, we need to evaluate the response to treatment by examining the parasites. Yeah, the fecal examination, the stool. That's it. But in vitro, we never do it. Like the microbial resistance test, no, we never do it. Maybe Professor Tomoyoshi may have experience in screening, the sensitivity screening for the any intestinal protozoa drug.
Yeah, I think for the patient-derived samples, like stool samples, if you luckily find entamoeba infections, but drug testing using the parasite from a stool sample is very difficult and reliable because of the presence of associated bacteria. Drug sensitivity is miscalculated and sometimes masked by the associated bacteria. So it's already available for the patient setting. It's not possible. You can test the sensitivity against the paromomycin or metronidazole of the entamoeba strains if you can establish the arsenic in a bacteria-free parasite cultures. I think it's very difficult at this stage. We have the parasite which is free from bacteria, but it's only a reference strains. We have only like a handful of the arsenic cultures from Japanese origin.
Okay, thank you very much. Very clear explanation. So next question from Alfian Wika from University Brawijaya for the second speaker. So he asks about the resistance in Plasmodium to the drug artemisinin and the other drugs. Several journals mention because of the change or mutation of drug target in the body of the parasite. So what about the drugs that target in PFDHODH and PFMQO as drug targets? Is it possible for resistance to occur and new compounds discovered? If possible, how about the mechanism that can occur and how to prevent this? Thank you, Prof.
Sure. Yeah. So it's a good question. It's our common understanding. Whatever the new drugs are made against Plasmodium, parasites are wise and smart enough to raise resistance to it. And they actually, even in the course of drug development, we also try to make a drug resistance in vitro. And this also helps us to identify the drug target. So when we in the process of making drug, we always, as I said, make a drug resistant line, and through a whole genome sequencing, we just sequence entire genome to identify mutations in the genome and try to understand what is the target of the drug. If we find the mutations in a specific gene, we can assume that is maybe somehow related. The mutation is somehow related to make the parasite resistance to the compound. So this is actually routine now for antimalarial drug developments. As you develop the drug, you also try to understand the drug resistant mechanisms and also mode of action. So, we have to be realistic. We have to keep making drugs in the future. And not a single, new drug is like a miraculous drug which can just cure the entire Plasmodium. It doesn't happen. So we need a second line, third line, and fourth line of drug in the future. That's a reality.
Thank you for thank you for your nice explanation. So I just want to ask a simple question to Professor Nozaki. So because of I am a pharmacologist, so I also interest to do some searching related to candidate drug for amoebiasis. So in your research, so you get the active compound from fungal. Is that true? Fungal, right? Yeah. So, but our lab is now searching from maybe from leaf, from extract or herbal compound. So, if we want to check the activity against anti-amoebasis, anti-entamoeba. So, can we do from extract of herbal, for example, water extract or ethanol extract or some herbal compound, and then go to the in vitro test. So it is possible or not? Or we have to find the active compound first?
Well, no, you can do either, both ways. Even without purifying the compound, you can directly test the crude extracts or maybe fractions in the animal model. That's okay, it's fine.
So in vitro model also can done, yeah, Professor. So
you can do it in vitro, like arsenic culture, and the second option would be like a hamster model, in vivo infection.
Oh, okay. So we can do in animal test.
Right.
Oh, okay. In rat or in some big animal, Professor. Animal?
We can also use a monkey, like a crab-eating monkey. But it's a bit laborious. So the first thing we should do is of course either hamster or inbred mouse strains such as C3H, Heston J, CVA mice. Yeah.
Okay. Okay. Thank you very much, Professor. So, is there any comment from the participant? Okay. Yeah.
I have one question in the chat.
Yeah. From Lucky Enggar Fitri. So regarding to the target on the DHODH enzyme, is there a possibility that a mutation has occurred in this target, and how to ensure that our drug candidate goes to parasite DOHDH, not to the human? Not to the human DHODH.
Yeah, of course, that's something we always have to make sure that the compound targets malaria enzyme, but not the human enzymes. So we always have like a counter assay. We first screen the compounds or libraries, extracts, whatever, against PF, DHODH. As a secondary or tertiary screening, we also do called counter state screening. So using human DHODH as a counter screening enzyme to make sure the compounds or extracts do not inhibit human counterpart. At the same time, we have to do a cell-based study. So using the compounds or extracts, we have to check whether the inhibitors kill the parasite, but not the human cell lines. Finally, we have to test it in animal models.
Okay,
so if the target is enzyme, and if the enzyme is present both in malaria and parasites and human, of course, we also need to do the counter screening. But if the target is the like a parasite-specific enzyme such as MQO, by the way, there is no MQO in human, and there is some enzymes are unique to the parasite, then that's sort of safe a target. Well, it's safe, but not completely safe. Because there is always off-target effect, so that you have to make sure, whatever the drug you are developing. So, yeah, but the rational target is specific enzyme, parasite-specific target.
Yeah, should be specific. Yeah.
And how about the mutation? How about the mutation? Because we have known that the parasite have a high mutation rate.
Yeah, so mutation occurs, as I explained. So whatever the drug we are testing, always a mutation happens. which makes a parasite resistant to the new compounds. Sorry, so what is the question? So mutation occurs. You mean the malaria?
So Luki, maybe
Yes,
Luki, can you?
yes. Is there any reports it might be about mutation in the HODH of the PF falciparum?
I see. I think there must have been. I don't recall the specific papers, but the DHODH based drug discovery has been going on for quite a long time. Now the leading compounds targeting DHODH is phase 2B trial, or it may be phase 3 already. So I'm pretty sure drug target is properly identified through both chemical studies as well as maybe structure studies, as well as the these mutational studies by whole genome sequencing. But I'm sorry, I really don't remember it.
Yeah. Thank you very much, Prof. Nozaki.
Okay. Hello.
Yes.
Hello. Next question for Professor Agnes.
Okay.
Is most person there is no harmful effect to the D. fragilis infection, but someone have infection in D. fragilis. Can you explain, Prof. Agnes, related to this kind of phenomenon?
Yeah, the Amoeba fragilis, some regarded it as a commensal, and I think maybe it is correct, but we don't know, yeah, because there are not many reported cases. But what we found, yeah, that this parasite, yeah, cause similar manifestation like amoebiasis, yeah. So the patients suffer from a persistent diarrhea with mucus, a lot of mucus, and then some with bloody diarrhea. And then it is not susceptible to metronidazole treatment. And then mostly with from the clinical symptoms, from the clinical manifestations, yeah, it is regarded very similar to amoebiasis, intestinal amoebiasis. However, yeah, it's not because we found that is the DNA amoeba fragilis. And then and it is surprising because it is present among the children less than 2 years of age. And at first we thought, yeah, that was amoebiasis among the children, among the babies. But how come babies contact, yeah, the infection, yeah. And then later on, we found that it's not an amoeba histolytica, but that is the amoeba fragilis. So I think it is very urgent, yeah, to differentiate between an amoeba histolytica infection and the amoebiasis, and I able to identify, yeah, to avoid the misdiagnosis and also the drug treatment. Because metronidazole is also toxic, especially if given so many times, yeah, repeatedly, and among the babies or children.
Okay, thank you very much, Prof. Agnes. Okay, so is there any further explanation from you? Okay, of course.
Yeah.
Of course, Professor. Yes.
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 Please. Yes.
I think Professor Agnes just show us a data of amazingly high seropositiveness among, I think this is the adult populations, 26 to 45 of age, 30% seropositive, you said. And what serological methods do you use for this?
That is the ELISA antibody, IgG anti-N amoeba histolytica. That is the common, diagnostic test for amoeba.
Using the crude antigen as on the plate. What antigen do you use?
So we bought the kit that is from the, I cannot remember, from the German made. What's the brand? And then it is already coated. Yeah, the plate is already coated with the antigen of an amoeba histolytica. And
I
then
see. I personally never used it myself.
Antibody. Yeah.
Okay. You know that there are a few different methods, such as gel diffusion precipitin test, like an Octaroni method, which is the least sensitive, but the most confirmed serological test. ELISA is more sensitive, but often pseudo-positive. So I'm just wondering, 30% positive in adult populations, how well that reflects infection state, status of the population. It's amazing amounts. And yet you don't see the parasite in the stool sample, but you see many amoebic liver abscesses patients. So this is sort of indirect evidence, but quite striking evidence that I have my colleagues at the Department of Parasitology at the university. Also, everybody say that amoebiasis is everywhere, but we have no proof that amoebiasis is really endemic in Indonesia until now. So I strongly encourage you to do like a stool PCR, because you have a very good way of typing of Blastocystis hominis, for instance. So you can use the same clinical samples for testing of anti-amoebic or anti-Dientamoeba fragilis PCR. So if you need some additional information, we can provide any information, so primer design, extraction methods. Yeah, that's all I'm saying.
Yeah. Yes, Prof. Nozaki. We have, the serology test we perform. We use two kits actually. One with ELISA, and then the other one with immunohem agglutination. It's a immunohem agglutination test. Yeah. So those two kits. And then, yes, I agree, because we are still collecting the samples. So those with diagnosed, SD and amoebiasis fragilis, and also amoebiasis, we will do the molecular typing as well to prove that it's not microscopically diagnosed, but also molecularly diagnosed. So I'm very glad that maybe we can, do a collaboration, and then for that kind of work, Thank you.
Okay, thank you for the explanation, Prof. Agnes and Prof. Nozaki. So there is another question from the participant for Professor Nozaki. So he asks about the clinical trial of fumagillin and ofalicin. Is that already start the human trial in this kind of drug?
No, I think in coming 4 or 5 years, I think we are going to make a series of fumagillin ofalicin derivatives for better efficacy and improve the safety. So basically, we're gonna rely on like animal experiments and chemical modifications at the first 3-4 years, and then we move on to the clinical trial, hopefully in Indonesia. And we have started contacting FDA, Indonesian FDA already, because there is no previous cases for registrations of the new anti-infective agent drugs in Indonesia yet. So we have to have a good discussions and conversation with FDA to know what sort of tests data are required for registration. So we have started it already. We are planning to start it, actually. We haven't really started it yet. So to answer the questions, maybe in 4 or 5 years, we are realistically think about the clinical study in humans. So first in humans.
Okay, very nice explanation. So related to the clinical trial that we'll conduct in Indonesia, so maybe we can doing like multi-center research, yeah, Professor. So because of we have a lot of patients in Indonesia, I mean to like a site of investigation, like that. So, but I'm not. I want to know about the exact mechanism of action of fumagillin and ofalicin. So it is different with the metronidazole, or maybe we can combine these two compounds with metronidazole.
Yes, thank you for asking the mechanism of actions. So as I briefly explained, ofalicin and fumagillin targets methionine aminopeptidase too. So the compounds inhibit translation. So the mode of action of the metronidazole is the forming nitro radicals, which was produced by pyruvate ferredoxin oxidoreductase, which is involved in energy production, energy synthesis, as well as redox regulation. So the target of metronidazole and target of the fumagillin ofalicin are totally different. So we hope that we can have, those two compounds in combination may have like a synergistic effects. So you're right, maybe the combination therapy might be really beneficial to consider in the future.
Okay, thank you very much, Professor. So, is there any question to the speaker from the audience? Maybe one question from to Professor Agnes. Prof. Agnes Dwi Penny from UNER asks about if there any molecular epidemiology of entamuba species in Indonesia.
Unfortunately, we haven't done it yet because
molecular epidemiology of entamuba.
Yeah. As far as I know, and also from our study, we haven't done the molecular epidemiology of Amoeba histolytica due to very scarce of samples we got. So not yet until now.
At the same time, it's the seropositiveness, because entamoeba, only entamoeba histolytica infections induces antibody productions. So if you have a positive serology, this is sort of indirect evidence for the presence of entamoeba histolytica infections. In general, and in the textbook, entamoeba dispar, entamoeba moshkovsky, should not have caused any seropositive conversion. So that's that's indirect evidence. But
that's indirect. Yeah, we know the prevalence, but is the epidemiology, the molecular epidemiology, maybe the type of an amoeba. So they want to know how many infected with an amoeba histolytica, how many with an amoeba dispar or Moskovsky, because what we identified, yeah, microscopically, that's the cyst, which is similar, yeah, between histolytica and dispar and on Moskovsky. Yeah, maybe, yeah, in future, if we have got enough samples, then we can do the typing. And there was a report, I know, yeah, from UNER, yeah, Airlangga University reported so high prevalence of amoebiasis, which was by microscopic examination. And I think, yeah, if you have positive samples like that, you can do the typing or molecular epidemiology for an amoeba. And it is interesting, yeah. So in Jakarta, it's very difficult we find amoeba, but if in the other cities, yeah, like in Surabaya or other cities or area in Indonesia, you got amoeba, yeah, you identify amoeba in the stool. So it is good if you isolate and then for further you do the typing, yeah, and that, so you can differentiate between an amoeba histolytica and dispar or Moskovsky and so on, yeah. So I think it is interesting to do it. Okay, that's all, Dr. Muli. Thank you, Prof. Tomoyoshi.
Okay, thank you very much. If there are no other questions, so everyone, finally we come to the end of this presentation. So we would like to say thanks to the all presenter for the interesting presentation and also to the participant for your active participation. So hopefully the presentation will be beneficial for everyone. Thank you very much. I will go back to Jordan, Dr. Jordan, to end this session.
Okay, thank you very much, Dr. Wawaimuli.
Your attention.
Okay, thank you very much, Dr. Wawaimuli, and thank you very much for the audience, and then also our honorable speakers, Tomoyoshi Sensei, and then Prof. Agnes, and then I think this session or this event or we can finish right now. And thank you very much for all of the participants, of the students and all of the audience. Thank you very much.
Thank you.
Audience, Adin.
Nice to meet you.
Okay. Okay, thank you.
Bye now.
Bye.
Bye-bye, Sensei. See you.
Thank you,
Bye now.
Sensei.
See you soon.
Thank you, Prof. Agnes.
Thank you, Prof.
Thank you, everybody.
Yeah,
Thank
you very much.
Makasih, Prof.
Arigato.
Yeah.
Arigato.
Yeah, thank you, Dr. Muli. Thank you, Prof. Agnes.
Sama. Yuk, bye-bye.
Ya.
Si Adin.
Terima kasih, Prof. Kami lagi lah.